Hello, and welcome to a rather short presentation on advanced cell model workflows. Today, we're going to be specifically talking about organoid identification, isolation, and transfer using an automated organoid picking instrument. So one thing we've realized over the last couple of years is that many organoid researchers will be doing their organoid work completely differently to one another. You could take 5 accounts doing brain organoids, for example, and each of these researchers in each of these institutes will be doing their work in a completely different manner to one another. However, most researchers will fall into one of 2 of these categories on the screen here. Either they'll be using live cell imaging as the endpoint assay, so they're preparing the sample, growing the organoids, picking the organoids either manually or using an automated organoid picker, adding the compounds or drugs of interest, and then identifying how these organoids grow and develop in presence of these compounds and drugs. On the other hand, the other researchers will be looking at molecular analysis as the endpoint assay instead. So again, using sample preparation, they'll be putting their organoids into an IncuCyte or a live cell imaging instrument, growing and developing them, then adding compounds or drugs to the whole range of organoids, then taking individual organoids for further downstream molecular analysis or gene expression or sequencing. So again, most people do things rather differently in the organoid world. Most researchers will fall into 1 or 2 of these categories. But what we're going to be talking about today is the boxes highlighted in red. The most suitable, quick, and gentle way of isolating and transferring individual organoids to essentially get 384 organoids into a plate and then add your drugs or compounds to identify how individual organoids grow and develop. So how do we do this? Well, we have an instrument at Sartorius, which is the Cell Selector Flex. And as you can see here in the short video, the instrument picks from a plate, as we're seeing here, and transfers the organoid into a destination plate. Now, the advantage of this is that the transfer is gentle, is reproducible, and you can identify exactly what you want to be picking up front. So you're picking just the organoids of interest that you'll be looking to transfer and then monitor thereafter. So moving on to the Cell Selector Flex, in a little bit more detail, what exactly is this instrument? Well, it comprises of 3 different parts. Firstly, an automated microscope with bright field phase contrast and epifluorescence. Then we have a robotic picking arm to basically pick the organoids of interest, move them from your sample plate onto your destination plate. And this can be done by either aspiration or scraping. And this is all controlled by a rather powerful image analysis workstation. So you're identifying the organoids you want to be picking. Let's say you want to have 96 organoids with 80 microns in diameter and perfectly spherical. You put this into your image analysis workstation. The instrument will then identify exactly what organoids you have in your source plate, pick them, and then transfer them. So in a little bit more detail, let's start on the top right-hand corner in terms of what the instrument is looking at. So we have an inverted microscope. There are 5 possible objectives ranging from 2x to 40x, bright field and phase contrast, as well as 5-channel fluorescence. What we're doing is we're adding our source vessel, so this could be a Petri dish or a 24-well plate or whatever source vessel you have in the lab to grow your organoids, adding this onto a motorized high-precision XY stage, monitoring with this inverted microscope. What we find in the source plate is then sent, the information is then sent to a retrieval arm here, which is then moving across, picking the organoid of interest, and then depositing into a number of destination plates here. Now, the deck tray is extremely important when it comes to organoid analysis. There are 2 possible deck trays available. The first one is a standard deck tray, which will warm up the sample up to 40 degrees. That's the standard configuration. Or, most interestingly for organoid research, especially when it comes to matrices like Matrigel, we also have a cooling option as well. So the deck tray can cool down to 4 degrees or warm up to 40 degrees. We'll explain the benefit and the advantages of that in subsequent slides. We also have 3 different picking arms available. Now, this is interesting when it comes to the organoid size you're picking. So as we can see here, the single cell picking tool, which is a glass capillary, ranges in diameter from 20 microns to 220 microns. This is generally used for non-adherent organoids in suspension. If, however, you have a very, very adherent colony, we can use the adherent colony picking tool, which is a stainless steel picking tip and ranges in size between 300 to 1,000 microns. And again, if you're picking, again, suspension organoids or something from a semi-solid media, we have plastic tips which range from 500 to 1,200 microns in diameter. So again, we'll be looking at single cells with a single cell picking tool, cell colonies, 3D structures you can pick using the glass capillary, stainless steel tips, or plastic disposable tips. Again, housing is extremely important, so there are two options available. We have the PCR HUD, which just offers basic user protection, but really for organoid research and workflows, the incubator flow box is probably the housing of choice. Here we have user protection as well as sterility. But the most important thing here is that we have three different environmental controls. We have temperature ranging from 4 degrees to 40 degrees, humidity control, and CO2 control. Now, all of these are extremely important in organoid research because if you're having an extended run, so you want to do picking for maybe 2, 3, 4 hours while you leave the instrument, go away, do something else, and come back to it, it's important that your cells and your organoids are kept in the best possible environment. And this is where the temperature, humidity control, and CO2 control come in very, very handy. Of course, one of the key advantages of cell-selective flex is that we can use pretty much any source vessel, so any cell culture plates with open top and clear flat bottoms, Petri dishes up to 100 millimeters, nanoarrays, pretty much any source vessel without a lid can be used. So you can grow your organoids in whatever you have in the lab. Generally, these can be picked from. And again, we can pretty much pick into any destination vessel as well. So cell culture plates up to 384 wells, Petri dishes, PCR tubes, strips, plates, multi-well slides, or custom plates. Again, probably not so relevant here, but source material, it picks pretty much everything and anything. Suspension cells, clones, adherent cells, colonies, clusters, clones in semi-solid media, 3D spheroids, and organoids. So pretty much any cell type or object can be picked using the cell-selective flex. It can also pick objects from fresh tissue, frozen tissue, or fixed tissue. So in general, there are 6 key advantages when it comes to organoid picking using the cell-selective flex. Number 1, this is an automated instrument. It does automated scanning, detection, and gating of your organoid structures. And this is based on either morphological and or fluorescence parameters. Essentially, if you put in, I would like to have 200 organoids, all 80 microns in diameter, all perfectly spherical, and all with a minimum of 10 microns away from the nearest neighbor, so we're not picking 2 or 3 organoids in one time. We put this into the software. The software says, yes, we have 95, 96 of these organoids, and then you can press go, and it will pick these automatically for you. Obviously, it only makes sense if the picking is gentle, and we have a wide range of organoid sizes. So organoids can range from pretty much any size, very, very small to very, very large, and this is what the cell selector allows. We have picking capabilities from 80 microns all the way up to 3.5 millimeters. And of course, the gentle picking is extremely key. There's not much point picking an organoid, transferring it, and then the organoid dying 2-3 days later. One of the key points again on that is that there are no changes in 3D structural morphology following picking and transfer. There's very, very little point picking an organoid and then a couple of days later realizing the structure or the morphology of the organoid has changed due to the picking. So we both have gentle picking, and because of the gentle picking, there are no changes in those 3D structural morphology of the organoid. Now, obviously, another key point is to make sure that when we're picking organoids, we have as low a volume of surrounding media as possible. This is important if we're injecting into something such as hydrogel or other liquid media, that we're basically taking as little of the source media as possible and transferring it, essentially just the organoid into a different matrix. We mentioned before about the cooled deck tray. This is extremely important when it comes to looking at objects or matrices such as matrigel. And the cooler the matrigel, the less likely it is to solidify. We all know the difficulties in matrigel work. As soon as it solidifies, or as soon as it gets to a little bit warmer, it begins to solidify, and then it becomes difficult to insert or deposit your organoid into the matrigel. The good thing about the cooled deck tray, it keeps the Matrigel extremely cold, so it doesn't solidify. It's very, very easy to put your organoid in without any air bubbles, and this can be done in plates with or without cell culture membranes. And finally, you need full documentation of the transferred organoids, right from the beginning of identifying exactly what parameters we're putting into the software, what organoids we're picking, the actual pick list itself, to what has been picked into which wells in the destination plate. This is fully documented by the instrument as well. We believe there are 6 key advantages to organoid research using an automated workflow such as this. So what is the overall process for generating 3D structures and transferring them into destination plates? It's actually very, very simple. What we're doing is picking from, as I mentioned before, pretty much any source material. So we have 4 different kinds of materials we can pick from. We can pick suspension organoids from ULA flat bottom surfaces. We can pick organoids from Matrigel domes or Matrigel layers. We can also pick organoids from standard ULA plates, or when there's something we're not going to really touch about in this presentation, picking organoids from third-party or cell-selector nanowell plates. Now, nanowell plates are essentially 6, 12, or 24 well plates with thousands upon thousands of small nanowells in larger macro wells. So 24 macro wells, up to 72,000 nanowells in cell line development, or over 1 million different nanowells if we're looking at antibody discovery or organoid research. On the right-hand side here, we have 3 examples of different plates where organoids have been seeded. Here we have a 6-well ULA plate. We can see there's many different organoids in this well, all different sizes, mostly spherical, but some more spherical than others. Here we have a 96-well ULA bottom plate with just a single organoid in the middle here. And then here we have organoids cultured in a ULA nanowell plate. Again, we can see the wells are much, much smaller, but again, individual organoids in each of these. So what we want to do, we've identified the organoids, we then want to start ranking, classifying, and gating these organoids based on whatever parameter you wish. So what we can see here on the right-hand side, we've got a number of different organoids here, and we can put in, for example, we would like all organoids just to be 80 microns plus or minus 1 micron in diameter. We want them to be as spherical as possible. We can put in those parameters up front. And we want them to be 10, 15, or 5 microns, for example, minimum distance away from the nearest neighbor, in order to prevent multiple organoids being picked up by the picking arm. So we can put all these parameters in. There's around about 150 different parameters that we can put in. So the idea is that you get just the organoid that you want at the end of your experiment in your destination plate. We don't want to be having organoids of different sizes, different morphologies, different shapes, different parameters in that destination plate. We would all want them to be exactly the same, or we can have a range of different organoids based on the parameters you put in. So first of all, generating the structures, then screening the structures based on the parameters you decide to add into the software. We can also add in an additional 3D matrix here. So we've mentioned and touched on before, the cooled deck tray option allows 2 destination vessel locations to be cooled or heated independently. And if we're looking at things such as Matrigel, this allows and maintains the Matrigel at around about 0 degrees. And when we do come to increase the temperature up to 40 degrees, of course, the Matrigel then begins to solidify. But the idea of the cooled deck tray is to keep that Matrigel at a nice cool level to facilitate organoid deposition into the Matrigel at that temperature. Then we heat it up to 40 degrees or whatever temperature we need to. That facilitates the optimal polymerization of the Matrigel. So there's a number of different ways and different advantages of this. One of the main advantages of using the cooled deck tray and doing this in an automated fashion is that we have 100% bubble-free deposition into hydrogel. And this is achieved by using controlled aspiration speeds, volumes, and destination vessel temperatures. Again, there's very, very little point of depositing a organoid into Matrigel if it's surrounded by, let's say, large amounts of source media or air bubbles. That's definitely something that we don't want to achieve. And this is actually helped by the cell selector flex that we don't achieve this. We get bubble-free deposition into Matrigel without any of those parameters. There are 2 different ways for achieving this transfer. Number 1 is aspirating the 3D structure from the source vessel and then dispensing into a destination vessel with hydrogel kept on this cooled deck tray. That's what we've described previously. The second way is to aspirate the hydrogel from a reservoir plate kept on the cooled deck tray then aspirate the 3D structure. So we have both the 3D structure and the hydrogel in, let's say, a glass capillary or plastic tip, and then dispense both into a destination vessel at the same time. Both approaches work perfectly fine on the cell selective flex. And as mentioned before, that low media volume, less than 1 microliter aspirated with a 3D structure, is absolutely critical in ensuring that we have optimal conditions for further growth and development. Here's a few examples of different organoids that we've collected over the years. We have human endometrial, intestinal, lung, retinal, colon, heart. We've looked at pretty much every single organoid there that can be picked. What we can see on the left-hand side, let's take the human intestinal organoid here, for example. We can see that was the organoid before picking. After picking, it's gone. One of the key advantages of the cell selective flex is that the organoid spheroid density is independent before picking. We can have as many organoids in the sample as we choose, and the instrument will identify each one to a high precision, then pick it, as we can see here. So we have a rather high density of organoids in this sample. I think this sample is embedded in matrix. We have before picking the organoid here. After picking, we can see the small ripples of the matrigel here. The organoid is gone without disturbing the surrounding organoids themselves. Obviously, this matrix independent. We can be picking from organoids in suspension. We can be picking from organoids in matrigel. It doesn't matter. Focal plane is also an important point, especially when we're coming to matrigel. And again, one of the key features of the cell selective flex, it is focal plane independence. We can be identifying this organoid here, picking that to a high degree of accuracy, or this organoid which is in a slightly different focal plane, or for example, this one which is even set further back. The instrument will identify the organoid to a high degree of accuracy and then pick it without disturbing surrounding organoids. And again, as we mentioned before, the preservation of morphology and structure following transfer is exceptionally important. Here we have a organoid, rather spherical, before picking from the source plate, and we can see on the destination plate there have been no changes in morphology or structure. We've also done additional downstream analysis to confirm this, and again, it's extremely important to make sure the organoid we have at the beginning is exactly the same structure and morphology as the organoid we have following transfer, and we have experimental evidence to show this. Finally, it makes sense to have a full compliance report based on every organoid that we've picked. So we have a GLP and GMP compliant documentation, which includes source ID, object ID, pick status, destination well ID, and 3D object metrics. All this is taken from your pick list and then can be exported via Excel after the experiment is completed. But again, we move from generating 3D structures, screening 3D structures, transferring the structures, and then we can generate this report at the end to ensure what we've picked is exactly where we've intended to pick it. So that's, again, a very, very brief overview into the organoid workflows. I believe one of my colleagues from the field application team will then move into a little bit more data on a number of different case studies, but open to questions just now.
Hello, everyone. My name is Alejandra Young. I am the Cell Selector FAS for Region 3 in North America. In this presentation, I will show you how the flexibility of the Cell Selector makes it possible to pick organoids from a variety of workflows. In order to do this, first, I will give you an overview of what makes the Cell Selector so flexible. And then I'm going to show you real-life cases in which we improve existing manual workflows or using the Cell Selector. As you saw in the presentation before, the Cell Selector Flex is a highly versatile picking system that can adapt to a wide variety of organoid workflows. The variety of picking tools and capillary sizes allows the picking and deposit of a range of organoid sizes, media types, and plate formats while ensuring that organoids remain viable and intact. The temperature controls on the deck tray with the heating and cooling options add to the flexibility with working with the matrigel. The automated software analysis as well as the click and pick selections ensure that the user can identify and isolate organoids meeting specific criteria. So now let's dive into the real-life cases. in case 1, the customer was transferring organoids in 2 steps. The first step, the organoids were cultured in 800 micrometer microwell plates containing iPSC medium. The organoid was transferred into a 96 well plate containing rinsing medium. And then in step 2, the organoid was transferred to a 24 well plate containing induction medium. So you can imagine this was labor intensive and time consuming, and it involved multiple manual handling steps. Also, the middle step here of rinsing was necessary because the customer will pick the organoid in a large volume of iPSC medium. So this step will allow the customer to rinse the organoid before transferring into the induction media, so that minimizes dilution and maybe also cross-contamination of that induction medium. With the cell selector, the goal was to fully automate this transfer of the organoids in one step from the microwell plate into the 96 well plate. So we use the feature of the cell selector that allow us to pick very low volumes. In this case, we were able to pick that organoid in 1 microliter of iPSC medium. So in that, we also use the feature of the cell selector in where the deck tray has a temperature control. So we kept the destination plate, the 96 well plate, at 37 degrees. This allows to keep those organoids very happy while we were picking them. And then also that increased the organoid viability. So as I told you, the microwell was very small, 800 micrometers, and also the organoids were small. So we decided to use the single cell picking module and the 220 micron glass capillary. We imaged with the 10x bright field, and we did a full automated analysis in that we screened for microwells containing organoids, and then we use different parameters to select the organoids and to filter them to then produce a picking list. On the right here I have a video to show you the cell selector in action. So you want to see the capillary going into the microwell and picking the organoid very gently in that 1 microliter of the iPSC medium. And then you can see how in the left, the cell selector software will give you these beautiful images of before and after picking. So you can do a quick QC while you are picking your organoids. So as we pick the organoids using the cell selector, the software automatically generate the before and after picking images. These images are saved as part of the experiment documentation. So you have a visual record showing that the organoid was successfully picked. Here we have a small organoid and image before and after picking that organoid. Here we have an example of a large organoid and the image before and after pick. In this slide, I wanted to show you the different size organoids transferred into the induction medium. And as you can see, the morphology of these organoids was intact. In summary, for this case, we successfully implemented one step fully automated workflows, saving time and labor. We also reduced transfer volume, eliminating the rinse step and saving reagents and consumables. We enhanced picking efficiency and organoid viability. In the second case I'm presenting here, the customer transferred organoids in 3 steps. And so in the first step, the customer will deposit droplets of 100% Matrigel into a 40 well plate. Then in the second step, the customer will pick one organoid from a 100% Matrigel layer that was deposited in a 12 well plate. Then that organoid was embedded in the previously set droplet in the 48 well plate. So as you can imagine, this was time and labor intensive to the manual organoid selection and Matrigel handling. Not to say the variability also that was involved here when delivering or embedding those organoids into that little drop in the 48 well plate. Using the cell selector, our goal was to fully automate the transfer of the single organoids from the Matrigel layer into that Matrigel dome deposit in the 48 well plate. So the organoids here were between 200 and 700 micrometers, so medium-sized organoids. Doing the full automation of this process eliminated the manual organoid and Matrigel handling. Also allow us to select the organoids based on morphology and size and ensure that the Matrigel domes were bubble-free, maintaining the organoid morphology. We also took the advantage of the deck tray temperature control and kept the destination plate at 22 degrees, so the whole plate was on a standard temperature. So when we deposit the droplet containing the organoid, they will polymerize at the same rate after touching the bottom. So since these were medium-sized organoids embedded in that 100% Matrigel layer, we decided to use the semi-solid picking module with the 500 micrometer disposable plastic tip. We imaged with the 2x bright field here, and we did automated image analysis to select the organoids by size and also by morphology. The customer here required us to transfer that organoid in a 10 microliter droplet. So to do that, we took advantage of the temperature controlling the deck tray of the cell selector, and we placed a buffer plate, a PCR plate, at 4 degrees. This PCR plate will contain the 100% Matrigel, so it was kept liquid by having it at 4 degrees. Then we placed the destination plate at 22 centigrades, and we kept it there because we wanted also to keep the temperature all throughout the plate constant. So to transfer that organoid in 10 microliters, what we did, we took 5 microliters of the liquid Matrigel, and then went and picked the organoid with another 5 microliters of the 100 Matrigel layer, and deposit that 10 microliter of volume containing the organoid into the empty destination plate. So one thing that we did here to ensure that there were no bubbles in the dome was to calibrate the deposit of this droplet. So we raised the tip to a space where we could deliver a very beautiful round droplet without any bubbles. Here I'm showing you the overview of the source plate with the Matrigel layer containing with the organoids in different sizes and different shapes. Also, you can appreciate here that some of those organoids are very close together. So this is something that we are very proud of. We can with the cell selector pick very precisely those organoids that are close without any disturbance of the ones that are nearby. And here I have a video for you showing you the semi-solid picking module with the plastic tick going into the matrigel layer and then picking that specific organoid. Again, the cell selector generates these beautiful images before and after while you're picking. Also, it's important to appreciate here that the matrigel layer is intact, there's no crack. It only shows the white spaces where we actually pick the organoids that we selected. I'm here also presenting more documentation for this experiment. On the left, I have a 25 organoids gallery that we selected for picking for this experiment. And on the right of that figure, we have the before and after image that were generated by the cell selector. You can see the last image here of the before and after show how precise can be the cell selector by picking the large organoid without disturbing the small one next to it. On the bottom, I just wanted to show you a drop, the droplet, the dome containing the organoid. Free of bubbles, so a very beautiful transfer here. To summarize, in this case, we were able to successfully automate the imaging selection and transfer of single organoids from a 100 matrigel layer into a 100 matrigel domes. So we save time and improve reproducibility and accuracy by eliminating a multi-step manual labor process, and we automatically identify organoids based on morphology and size. We ensure that the organoids maintain their morphology and that the matrigel domes remain bubble-free. In the last case that I'm going to present to you, the customer used a 3-step also to transfer organoids back from dome to dome. So in the first step, the customer will also deliver the droplets to the Fourier well plate, this time with 50 microliter matrigel. So then it will come to the source plate where he has these 3 domes in the center of the well and pick one organoid. And then in step 3, he will embed that organoid into the droplet that was already placed in the Fourier well plate. As you can imagine, again, time and labor-intensive manual organoid selection and transfer. And also here, the customer mentioned that he could only use Fourier well plates, and this really limit the throughput of the experiment. And this was because he couldn't deliver very small droplets, so he used 15 microliter matrigel volumes to generate these domes. So that was something that we wanted to be able to improve using the cell selector, just going from a 48 well plate to maybe a 96 well plate. So in this experiment using the cell selector, our goal was to fully automate the image and transfer of these organoids. And these organoids were actually GFP positive and RFP positive. They were 100 to 200 microns in size, and we really took advantage of all the features of the cell selector to be able to transfer these organoids in very little volume and also free of bubbles. So, to be able to deposit in smaller wells, not only 96 well plate, but we can go and deposit into 384 well plates. So in this case, we increased the throughput of the experiment by using 96 well plates instead of the 48 well plates. As I told you before, the organoids were small, 100 to 200 microns, so we decided to use the single cell picking module with the 220 glass capillary. We image at 4x bright field, GFP, and RFP, since we were multiplexing here. For the selection of organoids, in this case, we use both the automated image analysis and then the click and pick, which I am just going to show you in a minute how we use this feature from the cell selector. Again, here the customer wanted us to transfer the organoids in very little volume, and so to do this, we automated the whole organoid embedding into matrigel DOMs with the cell selector, and we were able to pick 4 microliters of the liquid 50% matrigel in the buffer plate, which was kept at 4 degrees, and then pick the organoid from the 25 microliter DOM with only 2 microliters of that 100% matrigel there. So we transferred that 6 microliter containing the organoid to the empty 96 wet plate kept at 22 degrees here. Here, I just wanted to show you the setup that we have for this experiment. So this is the cell selector here, deck tray with the target 1 and target 2. And on the far right here, we have the source plate. So in target 1, we placed the buffer plate, which was a PCR plate containing 50% matrigel. And then on target 2, we placed the destination plate, in this case, a 96 well plate. We kept this destination at 22 centigrades. And here I have a video just to show you the whole process. So here the capillary goes and picks the liquid matrigel, and then goes and picks the organoid, in this case in 2 microliters of that 100% matrigel there. So the 6 microliters go into the destination plate, kept at 22. So as soon as that droplet hits the bottom, it starts polymerization. And here I just wanted to show you the source plate. In this experiment, the customer wanted to transfer organoids that were GFP positive and RFP positive. So here I'm showing you the bright field channel, showing you 2 of those DOMs, 25 microliter DOMs in the source plate. And here the GFP positive expressors and the few that were RFP positive. So one thing that the customer wanted to achieve here was to make sure that the cell selector was able to differentiate between fluorescence, and also to see if we could pick the RFP organoids, which were had very little expression, and they have a lot of variability in size also. So that's what was one goal from this experiment. So here I'm showing you more documentation from the third case. On the left, I have both GFP positive and RFP positive organoids. In these galleries, specifically, the first column shows like a merge channel, then the bright field, following by the GFP channel, following by the RFP channel. As you can see, the GFP organoids only have the signal in the GFP channel, nothing in the RFP, and similarly the RFP only signal in the RFP, nothing in the GFP. On the right, I just am showing you one of the organoids that we pick. This was a GFP positive organoid. Again, on the right and the top, we're showing the bright field channel, in the middle the GFP, and on the right the RFP. On the bottom, we have the same organoid, just the images before and after, and the organoid is highlighted in green. So before you see the organoid, after you see the outline of that organoid we just pick, and the space where you just pick. So one thing to appreciate here is that when we pick that organoid, none of the neighbored organoids were disturbed. And here is a slide I wanted to have to show you how we use the click and pick feature of the cell selector. So in this case, I told you the organoids expressed in RFP they have very weak signal and they have different sizes. They were just a little bit, lots of variability in those organoids, but the goal of using click and pick was because the customer wanted to add some of those organoids of interest for him. So we use the click and pick, which is the feature where you look at the live image and then you use this crosshair over here in green, and with the mouse you are able to go and select whatever you are interested in adding to the picking list, and that's what we did here. So we added several of those RFP organoids to the list, and then we picked. And the last image here, from the documentation, this is just a picture that we took of those 6 microliter DOMs in the 96 well plate before we added the media. So on the right, we have that DOM containing the organoid, free of bubbles. So to summarize, here in this case 3, we successfully automated imaging selection and transfer of single GFP and RFP organoids, positive organoids, from 100 to 50% matrigel DOMs. We use click and pick to select additional organoids based on key criteria. We increase throughput and save time by switching to a 96 well destination plate. We reduce matrigel volume, conserving reagents, and we maintain organoid morphology and ensure bubble-free matrigel DOMs. As you can see, the cell selector is extremely flexible and adaptable to a wide range of workflows. With that, I thank you very much for your time, and I will be happy to take any questions. I cannot hear you.
Thank you, Ali. We have a few questions in the chat.
Okay, hi.
The first one is, why would I choose the Cell Selector Flex over manual or other automated picking systems?
So the Cell Selector Flex combines precision imaging with automated image analysis and cell retrieval, all in one instrument. So unlike manual picking, where you have biases to select what you want to collect, here we're using the software to allow us to screen thousands of cells or colonies with the specific parameters based on your specific criteria. So this is something that is very useful. The Cell Selector software allows you to see live imaging, picking, so you can actually do a quick QC of the picking while you're doing it. I will say also the system gives you like 3 instruments in one. You know, it's able to work with the single cell picking module, with the scraping module, with the semi-solid module. And in that sense, you can almost work with any application, any cell type, any cell, condition that you are used to work in your lab. So we can adapt the Cell Selector to any workflow in the lab. So I will say, that makes it a very valuable instrument to have in any laboratory.
Is that where the term flex comes from?
Yeah, so the flex will be, the 3 different modules, the capability of working with any platform, any plate format. Even if you are working with custom-made plates, we can adapt to that. So it's very flexible. It's like having 3 instruments in one, actually.
What kind of biological samples can it handle?
Again, unbelievable the amount of samples that we can handle. Sometimes, we say just anything you can see under the microscope, we can pick, but single cells, cell colonies, cells in suspension, adherent cells. We can work with any type of target in semi-solid media, like organoids, spheroids. We can even work with rare cells like CTCs. So very precise instrument. Also, using all the 3 modules, we can adapt really to any workflow.
And how does the imaging and detection part work?
Yeah, so the cell selector is equipped with an inverted microscope, and you want to be able to use bright field, phase contrast, all the fluorescence that you are used to work in a lab. We say we have like 6 channels, up to 14 colors that we can combine. So very flexible again with all these. I think that's pretty much it, what I want to say about that, yeah.
And then is the system gentle on the cells I pick? Will they survive and grow?
Yeah. So I think that's one thing that we are very proud of. Our system, we have the capability of controlling rate of aspiration, volumes. And so our system is very, very gentle, picking cells colonies. We have the possibility of doing optimization for each application. So we work on optimizing the perfect conditions to ensure high viability of colony cells, anything that we handle.